In a quiet laboratory in Scotland, bathed in the soft glow of fluorescent lights, a scientist gently cradles a lamb no bigger than a house cat. The year is 1996, and this isn't just any lamb, this is Dolly, the first mammal cloned from an adult cell, taking her first tentative breaths in a world that doesn't yet know how she'll change everything. Her wool is pristine white, her eyes bright with the ancient curiosity that belongs to all newborns. Yet genetically, she's identical to a sheep that lived years before her birth. It took 277 attempts to create Dolly, and scientists still can't explain why some cloning attempts succeed while others fail. Did you know that identical twins are nature's original clones, yet they develop completely different fingerprints? Or that there's an entire forest in Utah, 106 acres, of what appears to be separate trees? That's actually one massive cloned organism, possibly 80,000 years old. Tonight, as the world settles into sleep around us, let's embark on a gentle journey through the most fundamental mystery of existence: how life creates perfect copies of itself, and why those copies are never quite as perfect as they seem. Long before scientists ever dreamed of laboratories and microscopes, humans have been captivated by the idea of creating perfect duplicates. In the quiet corners of ancient libraries, scribes recorded stories that seem almost prophetic, now tales of beings crafted from clay, myths of gods breathing life into identical forms, legends of mirrors that could step forth from their frames as living beings. The ancient Greeks told stories of Pygmalion, who carved a statue so perfect that the gods brought it to life. In Jewish folklore, the golem represented humanity's desire to create life from lifeless matter, a creature of clay animated by sacred words. These weren't just stories. They were humanity's earliest explorations of a question that would echo through millennia. What if we could create life exactly as we wanted it? But nature, in her infinite wisdom, had already been answering this question for billions of years. Every time you've watched a spider plant send out its delicate runners, creating perfect little copies of itself, you've witnessed cloning in its most elegant form. When you've marveled at a field of strawberries, each plant connected to the next by gentle stems that carry identical genetic information, you've seen nature's own production line of perfect copies. The word clone itself comes from the ancient Greek klon, meaning twig or branch, those tender shoots that break away from parent plants to become identical offspring. It's a word that carries within it the gentle image of new life emerging from existing life, growth through careful separation, continuity through careful copying. What we call cloning today is simply the scientific refinement of processes that have been occurring naturally since the very first cells divided in ancient oceans. Every single-celled organism that splits to create two identical daughters is performing cloning. Every time your body heals a cut by creating new skin cells identical to the ones that were lost, it's engaging in the same fundamental process that scientists now harness in laboratories around the world. Step into any garden on a summer morning, when dew still clings to leaves and the air carries the sweet scent of growing things, and you're surrounded by nature's cloning masterpieces. The spider plant cascading from a hanging basket sends out long, graceful stems called stolons, each ending in a perfect miniature version of the parent plant. These babies dangle like green stars, genetically identical to their mother, waiting for the gentle touch of soil to begin their independent lives. In the soft light of dawn, strawberry patches reveal their own cloning secrets. What appears to be separate plants are often interconnected families, connected by runners that carry nutrients and genetic information between identical siblings. A single strawberry plant can create an entire patch of clones, each one capable of producing the same sweet, red fruit as the original. Beneath the soil, the stories become even more remarkable. Aspen groves that seem to contain hundreds of individual trees are often single organisms, massive clonal colonies connected by underground root systems. The Pando Grove in Utah, covering 106 acres and weighing an estimated 6,000 tons, is actually one gigantic organism that has been cloning itself for thousands of years. Walking through such a grove is like wandering through the branches of a single, impossibly vast tree. In tide pools, where ocean meets shore in gentle rhythms, sea anemones demonstrate another form of natural cloning. They can split themselves in half through a process called binary fission, creating two identical individuals from one. The process is so gradual and peaceful that watching it feels like observing a meditation on growth and renewal. The microscopic world reveals the most abundant cloning on Earth. Every moment, billions of bacteria are dividing through binary fission, creating identical copies of themselves with remarkable efficiency. In a single day, one bacterium can produce millions of identical offspring, each carrying the exact genetic blueprint of its parent. This process, so simple yet so fundamental, has been the backbone of life on Earth for over 3 billion years. Even in our own bodies, cloning happens constantly. Your skin replaces itself entirely every few weeks through a process of cellular division that creates identical copies of existing cells. When you cut yourself and watch the wound heal, you're witnessing your body's natural cloning abilities at work, creating new tissue that matches the old perfectly. Journey with me into the interior of a single cell, a world so small yet so intricate that it rivals any cathedral in its beauty and complexity. Here, in spaces measured in millionths of millimeters, the most fundamental act of cloning takes place every time a cell divides to create two identical daughters. Picture the cell nucleus as a precious library, its shelves lined with chromosomes that contain the complete blueprint for life. Each chromosome is like a carefully bound book, its pages written in the elegant script of DNA. Those famous double helixes that spiral like microscopic staircases, each rung a chemical letter in the longest story ever told. When a cell prepares to clone itself, it begins with one of nature's most meticulous processes, DNA replication. Imagine the double helix unzipping like the world's tiniest zipper, each strand serving as a template for creating its perfect partner. Molecular machines called DNA polymerases move along each strand like careful scribes, reading the genetic text and creating identical copies, letter by letter. This process is breathtakingly precise. In a human cell, over 3 billion base pairs must be copied perfectly, and the cellular machinery achieves an accuracy rate that would make any human copyist weep with envy. The few mistakes that do occur are usually caught and corrected by molecular proofreaders' specialized proteins that patrol the newly copied DNA like gentle editors, ensuring that the story of life is passed on without errors. The actual moment of cell division, called mitosis, unfolds like a carefully choreographed dance. The chromosomes condense and align themselves in the center of the cell, each one consisting of two identical copies joined at a point called the centromere. Protein fibers called spindles attach to each chromosome and begin pulling the identical copies toward opposite ends of the cell. As the chromosomes separate, the cell begins to pinch inward, like a soap bubble preparing to become two. The nucleus divides, creating two identical genetic libraries. The cytoplasm, that gel-like substance that fills the cell, distributes itself evenly between the emerging daughters. Finally, the cell membrane completes its gentle separation, and where there was one cell, now there are two, each carrying the exact same genetic information as the original. This process, repeated countless times throughout our bodies every day, is the foundation of all artificial cloning techniques. Scientists have learned to harness these natural cellular processes to convince cells to copy themselves in ways that create the remarkable cloned organisms that have captured our imagination and advanced our understanding of life itself. Our story of scientific cloning begins in the 1950s. In quiet laboratories, where researchers peered through microscopes at the tiny, translucent eggs of frogs floating in dishes of pond water. These scientists were asking a question that seemed almost philosophical. If every cell in an organism contains the same DNA, could any cell be coaxed to become an entirely new individual? John Gurdon, working at Oxford University, became captivated by this puzzle. In experiments that required the steadiest hands and the most patient heart, he began removing the nuclei from frog eggs, delicate operations performed under microscopes using glass needles finer than human hair. The eggs, stripped of their original genetic instructions, sat like empty pages waiting for new stories to be written. Then came the crucial step. Gurdon would carefully extract the nucleus from a cell taken from a frog's intestine, and with movements so precise they resembled the work of a watchmaker, transfer it into the empty egg. The process was so delicate that most attempts failed, but occasionally something magical would happen. The egg would begin to divide, creating first 2 cells, then 4, then 8, following the ancient rhythm of embryonic development. Some of these experiments produced tadpoles, tiny, swimming creatures that were genetically identical to the adult frog that had donated the intestinal cell. It was the first successful cloning of a vertebrate animal, though the world wouldn't fully appreciate the significance for years to come. Gurdon's work proved that cellular differentiation, the process by which embryonic cells become specialized as muscle, nerve, or skin cells, could be reversed. The 1960s and 1970s brought incremental advances, each experiment building on the last like stones in a careful wall. Scientists learned to work with different cell types to improve their techniques for nuclear transfer and to create the precise conditions needed for successful development. The work was painstakingly slow, with success rates measured in single digits. But each small victory brought researchers closer to understanding the fundamental mechanisms of cellular reprogramming. Throughout these decades, the researchers worked in relative obscurity. Their experiments were published in specialized journals, discussed at scientific conferences attended by dozens rather than hundreds, and funded by modest grants that barely covered the costs of frog eggs and laboratory equipment. They were driven by pure curiosity about the nature of life itself, with little inkling that their work would eventually lead to headlines around the world. The techniques refined during these early experiments, the delicate manipulation of microscopic cells, the precise timing required for successful nuclear transfer, the understanding of how cells could be reprogrammed to restart development would prove to be the foundation for everything that followed. Every cloned animal that would capture the world's attention in later decades owed its existence to the patient scientists who spent their careers learning the secrets of frog eggs floating in laboratory dishes. On a quiet morning in July 1996, in a laboratory at the Roslin Institute in Scotland, something extraordinary stirred in a small incubator. Among the glass chambers and monitoring equipment, a newborn lamb took her first breaths, unaware that she was about to change the world's understanding of what was possible in science and life itself. Her name was Dolly, chosen by the researchers in honor of country music star Dolly Parton, and she appeared to be perfectly ordinary white wool, bright eyes, the characteristic bleating of a healthy newborn sheep. But Dolly carried within her cells a story that was anything but ordinary. She had been created not through the natural union of sperm and egg, but through a process that seemed to belong more to science fiction than to the rolling hills of Scotland. The process that created Dolly began with a single cell taken from the mammary gland of a 6-year-old Finn Dorset sheep. This cell, which had lived its entire existence as part of breast tissue, contained the complete genetic blueprint needed to create an entire sheep. But like all adult cells, it had settled into its specialized role and forgotten how to become anything else. Dr. Ian Wilmot and his team had spent years perfecting the technique that would convince this mammary cell to remember its embryonic potential. They carefully extracted the nucleus from the mammary cell and prepared it through a process called cell cycle synchronization, essentially putting the cell into a state of genetic hibernation where its DNA was ready to respond to new instructions. Meanwhile, they prepared an egg cell from a Scottish blackface sheep, removing its original nucleus to create a blank slate for development. The marriage of these two cells, the nucleus from the Finn Dorset mammary cell and the prepared egg from the Scottish blackface, required a precisely timed electrical pulse that fused them together and awakened the dormant developmental program within the transferred DNA. Out of 277 attempts, only 29 embryos developed to the point where they could be implanted into surrogate mothers. Of these, only one pregnancy proceeded to full term, resulting in the birth of Dolly. The success rate was extraordinarily low, but the single success was enough to prove that adult mammals could indeed be cloned from differentiated cells. Dolly lived a relatively normal sheep life at Roslin, grazing in Scottish pastures and eventually becoming a mother herself through natural reproduction. She gave birth to 6 lambs over her lifetime, proving that cloned animals could reproduce normally. Her offspring were conceived naturally and carried no unusual genetic markers. They were simply the children of a very special mother. The announcement of Dolly's existence in February 1997 sent shockwaves around the world. Newspaper headlines proclaimed the dawn of a new age, ethicists debated the implications, and the public grappled with questions that had previously existed only in the realm of science fiction. President Clinton ordered a moratorium on federal funding for human cloning research. The Vatican issued statements about the sanctity of human life, and science fiction movies suddenly seemed much less fictional. Dolly herself remained blissfully unaware of the controversy swirling around her. She lived at Roslin for 7 years, becoming something of a celebrity and inspiring countless visitors who came hoping to glimpse the world's most famous sheep. When she died in 2003 from a lung tumor, her body was preserved and is now displayed at the Royal Museum of Scotland, where visitors can still see the gentle creature that opened the door to modern cloning. In the hushed atmosphere of modern cloning laboratories, scientists work with tools so precise they can manipulate individual cells with the delicacy of a master calligrapher forming perfect letters. The process of nuclear transfer that created Dolly has been refined into an art form that combines cutting-edge technology with techniques requiring almost supernatural steadiness of hand. The laboratory where nuclear transfer takes place resembles a high-tech sanctuary. Specialized microscopes equipped with micromanipulators allow scientists to work at magnifications where a single cell fills their entire field of view. The micromanipulators themselves are marvels of engineering mechanical arms that can translate the relatively gross movements of human hands into movements measured in thousandths of millimeters. The process begins with the careful selection of donor cells. These might be skin cells from a prize-winning racehorse, tissue samples from an endangered species, or cells that have been genetically modified to produce valuable proteins. The cells are cultured in sterile conditions, growing in dishes filled with nutrient media that provides everything they need to remain healthy and viable. Meanwhile, egg cells are harvested from female animals through procedures that have become increasingly refined and humane. These eggs represent nature's most sophisticated developmental machinery, cellular environments, perfectly designed to nurture the transformation of a single fertilized cell into a complete organism. The eggs are treated with the same care as precious gems, maintained at exact temperatures and in precisely balanced chemical solutions. The actual nuclear transfer requires skills that take years to master. Using a glass pipette finer than human hair, the scientist approaches the egg cell and carefully removes its nucleus, the command center that contains the egg's original genetic instructions. This enucleated egg now sits empty, retaining all of its developmental machinery, but lacking the genetic blueprint needed to create life. Next comes the delicate introduction of the donor nucleus. The donor cell is positioned next to the enucleated egg, and the two are fused using either electrical pulses or chemicals that temporarily make cell membranes permeable. When successful, the donor nucleus finds itself in an entirely new environment, surrounded by the egg's cytoplasm, which contains all the molecular signals needed to reprogram adult cells back to an embryonic state. This reprogramming process is perhaps the most mysterious aspect of cloning. The egg cytoplasm contains factors that can essentially erase the cellular memory of the donor nucleus, stripping away the molecular markers that identified it as a skin cell or liver cell, and returning it to a state where it remembers how to become any cell type in the body. It's as if the egg whispers ancient instructions to the nucleus, reminding it of developmental pathways it had forgotten. Not every nuclear transfer attempt succeeds. The process is still inefficient, with success rates varying depending on the species, the age and type of donor cells, and the skill of the technician. Many embryos fail to develop properly, stopping their growth at various stages. Those that do develop successfully are carefully monitored as they progress through the early stages of embryonic development. The embryos that show normal development are then implanted into surrogate mothers, where they will hopefully continue developing until birth. The surrogate mothers receive the same careful prenatal care as those carrying naturally conceived offspring, with regular monitoring to ensure both mother and developing clone remain healthy. Throughout this process, scientists maintain detailed records of every step, analyzing success rates, identifying factors that contribute to successful cloning, and continuously refining their techniques. Each cloning laboratory becomes a repository of hard-won knowledge about the intricate dance between donor cells, egg cytoplasm, and the mysterious processes that govern early development. Step into any commercial greenhouse on a spring morning, and you're surrounded by one of the most successful applications of cloning technology in human history. The rows of identical plants stretching into the distance, perfect tomatoes, uniform roses, consistent strawberries, represent decades of refinement in plant cloning techniques that have revolutionized agriculture and horticulture. Plant cloning, or vegetative propagation, takes advantage of plants' natural ability to regenerate entire organisms from small pieces of tissue. Unlike animals, which require complex procedures to achieve cloning, many plants can be convinced to grow from single cells with relatively simple techniques. A small cutting placed in the right growing medium can develop roots and shoots, becoming a genetic copy of its parent plant. In tissue culture laboratories, scientists work with plant material at the cellular level. Tiny pieces of plant tissue, sometimes just a few cells thick, are placed in sterile containers filled with carefully balanced nutrients. These cultures are maintained in climate-controlled rooms where temperature, humidity, and lighting are precisely regulated to encourage growth. Under these conditions, a single piece of leaf tissue can give rise to dozens or even hundreds of identical plants. The advantages of plant cloning become immediately apparent when you consider crop production. A single superior plant, one that produces exceptional fruit, resists disease particularly well, or adapts perfectly to local growing conditions, can be propagated into millions of identical copies. This consistency allows farmers to predict exactly how their crops will perform, when they'll ripen, and what qualities the final product will possess. Many of the fruits you enjoy are actually clones. Every Granny Smith apple shares the same genetic heritage, traced back to a single tree discovered in Australia in the 1860s. The bananas in your local grocery store are genetic copies of plants selected for their superior taste, texture, and shelf life. Wine grapes are propagated as clones to maintain the precise characteristics that define different varietals and vintages. The nursery industry relies heavily on cloning to produce the ornamental plants that grace our gardens and homes. That perfect rose bush, with its ideal flower shape and color, exists as thousands of identical copies because it was cloned rather than grown from seed. Fruit trees are often grafted, a form of cloning where branches from superior trees are attached to different rootstock, creating orchards of genetically identical trees. Conservation efforts also benefit tremendously from plant cloning techniques. When species become endangered or extinct in the wild, tissue culture can preserve their genetic heritage indefinitely. Seeds may lose viability over time, but plant tissues can be maintained in culture for decades, ready to be regenerated into full plants when conditions allow for reintroduction. Some of the most remarkable conservation successes involve plants that were reduced to just a few individuals in the wild. The California fan palm, various orchid species, and numerous medicinal plants have been preserved through cloning programs that maintain genetic diversity while building populations large enough to ensure species survival. Research applications of plant cloning continue to expand our understanding of plant biology. Scientists can create thousands of identical plants for experiments, eliminating genetic variation as a confounding factor in their studies. This precision allows researchers to test the effects of different growing conditions, treatments, or genetic modifications with unprecedented accuracy. The speed of plant cloning also offers advantages for research and production. While growing plants from seed might take months or years to produce mature specimens, cloning can sometimes produce viable plants in weeks. This acceleration allows plant breeders to work through multiple generations quickly, speeding the development of new varieties with desired characteristics. In the quiet corridors of medical research facilities, scientists work on applications of cloning technology that could revolutionize how we treat disease and injury. This field, known as therapeutic cloning, focuses not on creating complete organisms, but on producing specific cells and tissues that could be used to heal and repair the human body. The concept begins with the same nuclear transfer techniques used to create Dolly, but instead of allowing the cloned embryo to develop into a complete animal, researchers harvest it at a very early stage to obtain embryonic stem cells. These remarkable cells possess two unique properties. They can divide indefinitely in laboratory culture, and they retain the ability to become any cell type in the human body. Imagine being able to treat a patient's heart attack by injecting them with healthy heart muscle cells grown from their own genetic material. The cells would be perfect genetic matches, eliminating the risk of rejection that plagues current transplant procedures. This is the promise of therapeutic cloning, personalized medicine taken to its ultimate conclusion. Current research focuses on several major applications. For patients with Parkinson's disease, whose symptoms result from the death of specific brain cells that produce dopamine, therapeutic cloning could provide replacement neurons grown from the patient's own cells. Diabetics could receive pancreatic cells capable of producing insulin. Spinal cord injury patients might benefit from replacement nerve cells that could restore damaged connections. The process of creating these therapeutic cells begins in specialized laboratories, where scientists have learned to guide stem cell development with remarkable precision. By adding specific chemical signals to cell cultures, researchers can encourage stem cells to develop into particular cell types. The culture dishes become like molecular nurseries where cells are gently coaxed along specific developmental pathways. Growing tissues and organs presents even greater challenges and possibilities. Scientists are learning to create three-dimensional scaffolds, framework structures, that provide support and guidance for growing tissues. These scaffolds can be made from naturally occurring materials or synthetic polymers that gradually dissolve as natural tissue grows to replace them. Some of the most promising current research involves creating organoids simplified versions of organs that can be grown in laboratory dishes. These miniature organs don't have the full complexity of natural organs, but they can provide valuable insights into disease processes and serve as testing grounds for new treatments. Heart organoids beat with regular rhythms. Brain organoids show electrical activity characteristic of neural tissue. And liver organoids can perform many of the metabolic functions of natural liver tissue. The field has already achieved some remarkable successes. Researchers have grown sheets of skin cells for treating burn patients, created cartilage for repairing joint damage, and developed techniques for growing blood vessels that could be used in cardiac surgery. While these applications don't all require cloning technology, they demonstrate the potential for using cellular engineering to address medical challenges. Recent advances in induced pluripotent stem cell, iPSC, technology have provided an alternative to traditional therapeutic cloning. Scientists have learned to reprogram adult cells directly back to an embryonic-like state without using eggs or creating embryos. This approach addresses some of the ethical concerns associated with therapeutic cloning, while potentially providing the same medical benefits. The regulatory landscape for therapeutic cloning varies significantly around the world. Some countries have embraced the research while maintaining careful oversight. Others have imposed restrictions based on ethical considerations, and still others are developing frameworks for evaluating these technologies as they mature. The goal is to balance the tremendous potential benefits against legitimate concerns about the use of human embryos in research. Clinical trials of therapies derived from cloning and stem cell research are beginning to show promising results. Patients with certain types of blindness have received injections of retinal cells derived from stem cells, with some showing improved vision. Trials for heart disease, spinal cord injury, and various other conditions are in various stages of development. Since Dolly's groundbreaking birth, laboratories around the world have become modern arks, housing an increasingly diverse collection of cloned animals that reads like a roll call from Noah's vessel. Each species presents its own unique challenges and teaches scientists something new about the intricacies of reproduction and development. Cats entered the cloning world in 2001 with the birth of CC, short for carbon copy, a kitten born at Texas A&M University. Interestingly, CC looked quite different from her genetic donor due to the random inactivation of X chromosomes that creates the distinctive patterns in female cat coats. This discovery helped scientists understand that cloning doesn't produce perfect physical copies. Environmental factors during development can still create unique individual characteristics. The cloning of horses has captured particular attention due to their value in racing and breeding. Prometea, born in Italy in 2003, was the first cloned horse, followed by numerous others including polo ponies and racing champions. The horse cloning industry has developed around preserving the genetics of exceptional animals, allowing their genetic legacy to continue even after death. Dogs proved to be one of the most challenging species to clone successfully. Snuppy, an Afghan hound born in South Korea in 2005, was the first successfully cloned dog, but the process required hundreds of attempts. Dogs have unusual reproductive cycles and embryonic development patterns that make nuclear transfer particularly difficult, requiring researchers to develop specialized techniques. The cloning of cattle has become almost routine in some agricultural applications. Cloned cows can produce identical twins separated by years, allowing farmers to replicate animals with particularly desirable traits such as high milk production or superior meat quality. The technology has also been used to preserve the genetics of prize-winning bulls and championship breeding stock. Pigs represent one of the most medically significant cloned species due to their physiological similarity to humans. Cloned pigs have been genetically modified to produce organs that might be suitable for transplantation into humans, addressing the critical shortage of donor organs. Other cloned pigs have been engineered to produce human proteins in their milk, or to serve as models for human diseases. The cloning of endangered species has captured public imagination and offers real hope for conservation efforts. Scientists have successfully cloned several endangered species, including the gaur, a type of wild ox, the banteng, a Southeast Asian wild cattle species, and the African wildcat. While these efforts face significant challenges, they demonstrate that cloning could serve as a tool for preserving genetic diversity in threatened populations. Perhaps most remarkably, scientists have begun attempting to clone extinct species using genetic material preserved in museums or recovered from frozen remains. While no extinct mammal has yet been successfully cloned to adulthood, advances in genetic engineering and cloning technology are bringing this possibility closer to reality. The Woolly Mammoth Project, which aims to modify elephant embryos with mammoth genes, represents the cutting edge of this de-extinction research. Pet cloning has emerged as a commercial service, though it remains expensive and controversial. Companies now offer to clone beloved cats and dogs for owners willing to pay tens of thousands of dollars. While the cloned pets share the same DNA as the originals, they often have different personalities and physical characteristics, leading to complex emotional responses from owners who expected perfect replicas of their departed companions. Each cloned animal teaches scientists something new about species-specific reproductive biology. The success rates, gestation periods, and developmental challenges vary dramatically between species, requiring researchers to develop customized techniques for each type of animal. Some species, like sheep and cattle, clone relatively easily, while others, like primates, have proven extraordinarily difficult to clone successfully. The health and longevity of cloned animals has been a subject of ongoing study. Early concerns about accelerated aging and health problems have been largely allayed as cloned animals have lived normal lifespans and shown typical health profiles. Dolly herself lived to be 6 years old, shorter than the typical sheep lifespan, but her death was attributed to a lung infection rather than problems related to cloning. The welfare of cloned animals remains a priority for researchers and regulatory agencies. Cloning procedures are subject to the same animal welfare regulations as other research activities, and scientists work to minimize any stress or discomfort associated with the procedures. Surrogate mothers receive careful veterinary attention throughout pregnancy, and cloned offspring are monitored closely for any signs of health issues. As we settle deeper into our exploration of cloning, it's time to gather around the metaphorical fireplace and gently examine some of the profound questions this technology raises about identity, individuality, and what it means to be human. These aren't questions with simple answers, but rather contemplations that invite us to think more deeply about ourselves and our relationship with the natural world. The question of identity in cloning often begins with a simple thought experiment. If someone created a genetic copy of you, would that person be you? The answer, science tells us gently, is both yes and no. Genetically, a clone shares your complete DNA blueprint. But as we've learned from identical twins' natures, own clones' genetics alone doesn't determine who we become. Consider the story of identical twins separated at birth who grow up in different environments. While they might share surprising similarities in preferences, talents, and even mannerisms, they develop into unique individuals with their own personalities, memories, and relationships. A clone would be similar, genetically identical, but shaped by different experiences, different timing of birth, and different environmental influences during development. The philosophical tradition has long grappled with questions about the nature of personal identity. Are we our memories, our bodies, our consciousness, our relationships. Cloning doesn't create new philosophical problems so much as it highlights questions that philosophers have been considering for centuries. What makes you, you isn't just your genetic code, but the complex interplay of biology, experience, choice, and chance that shapes every human life. From a spiritual perspective, different traditions approach cloning with varying degrees of acceptance and concern. Some see it as humanity working in partnership with divine creative forces, using the tools of science to preserve and protect life. Others worry about humans overstepping appropriate boundaries, attempting to play roles traditionally reserved for higher powers. Most religious perspectives acknowledge both the potential benefits and the need for careful ethical consideration. The concept of the soul enters these discussions with gentle complexity. If souls exist, do clones have them? Most theological perspectives suggest that souls aren't tied to genetics, but rather to the individual consciousness that develops in each person. A clone would be understood to have their own soul, their own spiritual journey, and their own relationship with the divine, regardless of their genetic origins. Cultural perspectives on cloning vary significantly around the world, reflecting different values about family, individuality, and the role of technology in human life. Some cultures place great emphasis on genetic lineage and might view cloning as a way to preserve important family traits. Others prioritize individual uniqueness and might see cloning as potentially diminishing human diversity. The question of consent in cloning raises interesting ethical considerations. Unlike natural reproduction, where genetic material from two people combines in ways that create new combinations, cloning uses the complete genetic blueprint of one person. Should that person have the right to control how their genetic material is used? What obligations might they have to resulting clones? Concerns about commodification, treating human life as a product to be manufactured, represent one of the most serious ethical challenges raised by cloning technology. The worry isn't just about the clones themselves, but about how society might begin to view human reproduction and human value if cloning becomes commonplace. The diversity argument suggests that human genetic diversity serves important evolutionary and social purposes. If cloning became widespread, particularly if people preferentially cloned individuals considered to have superior traits, it might reduce the genetic diversity that helps our species adapt to changing conditions and challenges. Interestingly, many of the ethical concerns about human cloning assume a level of predictability about genetic outcomes that science doesn't actually support. We now understand that gene expression is heavily influenced by environmental factors, that development involves considerable randomness, and that even identical genetic codes can produce surprisingly different outcomes. The precautionary principle suggests that when dealing with technologies that could have significant consequences, we should proceed carefully and err on the side of caution. This principle has guided much of the policymaking around human reproductive cloning, leading most countries to prohibit it while allowing research into therapeutic applications. Perhaps most fundamentally, cloning invites us to consider what we value about human reproduction and family relationships. Natural reproduction involves elements of chance, surprise, and the blending of traits from two people who choose to create new life together. Cloning, by contrast, involves deliberate choices about creating specific genetic combinations. Neither approach is inherently superior, but they represent different relationships between human intention and natural processes. The relationship between cloning and science fiction reads like a conversation between imagination and reality that has been ongoing for nearly a century. Long before scientists could actually clone mammals, writers and filmmakers were exploring the possibilities, fears, and wonder that this technology might bring to human experience. Early science fiction often portrayed cloning as a tool for creating armies of identical soldiers or workers, perfect copies, that could be mass-produced for specific purposes. These stories reflected mid-20th century anxieties about industrialization and conformity, imagining futures where human uniqueness might be sacrificed for efficiency and control. The reality of cloning has proven far more nuanced and far more respectful of individual dignity. The 1970s brought more sophisticated explorations of cloning in fiction, often focusing on themes of identity and what makes us human. Movies like The Boys from Brazil explored the frightening possibility that cloning might be used to recreate dangerous historical figures, while other works examined the psychological impact of discovering one's cloned nature. These stories help the public grapple with philosophical questions about genetics and identity before the technology became reality. Popular culture's portrayal of cloning has often emphasized the dramatic possibilities while glossing over the technical realities. Movie clones are frequently portrayed as instantly adult copies with all the memories and personality of their originals, something that bears no resemblance to actual cloning, which produces normal infants that develop naturally over time. The clone army trope that appears throughout science fiction assumes a level of control over personality and behavior that genetics simply doesn't provide. Real cloning produces individuals with the same genetic potential as their donors, but personality, skills, and choices develop through experience and can't be predetermined or controlled through genetic manipulation alone. Some science fiction has explored more realistic scenarios, examining how cloning might be used for medical purposes or conservation efforts. These stories often focus on the ethical dilemmas and unintended consequences that might arise from well-intentioned uses of cloning technology, providing thoughtful explorations of issues that scientists and ethicists continue to grapple with today. The gap between fiction and reality becomes particularly apparent when considering timelines. Science fiction often portrays cloning as a quick process that can produce adult copies in days or months. Real cloning follows the same developmental timeline as natural reproduction. Cloned mammals must grow and develop over the same months or years as naturally conceived individuals. Memory transfer, a common theme in cloning fiction, remains pure fantasy. Memories are stored in neural connections that develop through experience, not in genetic code. A clone of any person would have to learn language, develop relationships, and acquire knowledge through the same processes as any other individual. The portrayal of clones as somehow less than fully human has been one of the most troubling aspects of science fiction's treatment of cloning. This portrayal has no basis in scientific reality. Clones are genetically identical to their donors and would be fully human in every meaningful sense. Such fictional portrayals may contribute to stigmatization and misunderstanding about the nature of cloning. More recently, science fiction has begun exploring cloning with greater nuance and scientific accuracy. Stories now examine the social and psychological implications of cloning technology while respecting the full humanity of cloned individuals. These works often focus on themes of family relationships, medical applications, and conservation efforts that more closely mirror real-world cloning research. The influence flows both ways. Science fiction has inspired some researchers to pursue cloning technologies, while real scientific advances continue to inform fictional portrayals. This dialogue between imagination and reality has helped society process the implications of cloning technology and consider how it might be used responsibly. Current science fiction often explores what-if scenarios that help us think through the implications of different policy choices around cloning. Should therapeutic cloning be widely available? How might society change if cloning became routine? What safeguards might be needed to prevent misuse? These fictional explorations serve as thought experiments that can inform real-world decision-making. The persistence of certain science fiction tropes has sometimes hindered public understanding of cloning. Surveys show that many people still believe clones would be identical copies with the same memories and personalities as their donors, or that cloning could be used to achieve immortality. Science education efforts must continually work to distinguish between fictional portrayals and scientific realities. In research institutions around the world, the daily work of cloning has settled into rhythms that would surprise those who imagine it as high-drama science fiction. The reality is quieter, more methodical, and focused on solving specific problems rather than creating sensational breakthroughs. These modern laboratories represent the maturation of cloning, from experimental curiosity to practical tool. Agricultural research facilities use cloning to develop crops that can feed growing populations, while using fewer resources and withstanding climate change. Scientists work with plant tissues in sterile culture rooms, creating varieties that might resist drought, produce higher yields, or contain enhanced nutrition. The work involves careful observation, meticulous record-keeping, and the patience to nurture experimental plants through multiple generations. Conservation laboratories maintain frozen repositories of genetic material from endangered species, modern genetic libraries that preserve biological diversity for future generations. Technicians work with liquid nitrogen storage systems, maintaining cell cultures and tissue samples at temperatures that effectively stop biological time. When species recovery efforts require genetic diversity that no longer exists in wild populations, these repositories provide irreplaceable resources. Medical research facilities focus increasingly on therapeutic applications of cloning technology. Scientists culture stem cells derived from cloning procedures, guiding their development into specific cell types that might treat degenerative diseases. The work requires extraordinary precision and sterile technique, as even minor contamination can destroy months of careful cell culture work. Veterinary cloning services have emerged to serve both commercial agriculture and pet owners seeking to preserve the genetics of valued animals. These facilities combine the technical expertise developed in research settings with the practical considerations of serving clients who want specific outcomes within reasonable timeframes and budgets. Quality control in modern cloning facilities rivals that found in pharmaceutical manufacturing. Every step of the process is documented, from the selection of donor cells through the birth and health monitoring of cloned offspring. This documentation serves both scientific and regulatory purposes, ensuring that procedures meet established standards and that results can be reliably reproduced. The daily rhythm of a cloning laboratory involves cycles of preparation, execution, and monitoring that can extend over months. Cell cultures must be maintained with regular feeding and monitoring. Nuclear transfer procedures require scheduling that coordinates the availability of donor cells, recipient eggs, and skilled technicians. Embryo development must be carefully tracked through critical developmental milestones. Training programs for cloning technicians have become increasingly sophisticated, combining theoretical knowledge with hands-on experience in the precise manipulations required for success. New technicians spend months learning to handle individual cells, to judge embryo quality, and to perform nuclear transfer procedures with the consistency required for reliable results. International collaboration has become routine in cloning research, with scientists sharing techniques, comparing results, and coordinating efforts to address common challenges. Professional conferences bring together researchers from around the world to discuss advances, troubleshoot problems, and plan collaborative projects that might be too large or complex for individual laboratories. Regulatory oversight has evolved to match the maturation of cloning technology. Agencies responsible for animal welfare, food safety, and medical device approval have developed specific protocols for evaluating cloning procedures and their products. This regulatory framework provides guidance for researchers while protecting public safety and animal welfare. The economics of cloning have shifted as the technology has matured. Early cloning experiments were funded primarily through research grants and were justified by their contribution to scientific knowledge. Current cloning activities increasingly involve commercial applications where success is measured by practical outcomes rather than just scientific advancement. Modern cloning facilities must balance multiple priorities: advancing scientific knowledge, meeting commercial objectives, maintaining animal welfare standards, and satisfying regulatory requirements. This complexity requires careful planning and coordination between scientists, business managers, veterinarians, and regulatory specialists. As we look toward the horizon of cloning technology, the future appears filled with possibilities that could gently transform how we approach medicine, conservation, and our relationship with the natural world. These emerging applications focus not on the dramatic scenarios of science fiction, but on practical solutions to real problems that affect human welfare and environmental health. Precision medicine represents one of the most promising frontiers for cloning technology. Researchers envision a future where patients could receive tissues and organs grown from their own cells, eliminating the risk of rejection and reducing dependence on immunosuppressive drugs. This personalized approach to treatment could revolutionize organ transplantation, making it safer and more effective while dramatically reducing waiting times for patients who need life-saving procedures. The development of disease-specific cell lines through therapeutic cloning could accelerate research into conditions like Alzheimer's disease, Parkinson's disease, and multiple sclerosis. Scientists could create cellular models of these diseases using cells from affected patients, allowing them to study disease progression and test potential treatments with unprecedented precision and relevance. Agricultural applications of cloning may help address global food security challenges, as climate change and population growth strain traditional farming systems. Researchers are developing crops that could thrive in harsh conditions, plants that resist drought, tolerate salt water, or grow in degraded soils. These advances could help farmers maintain productivity even as environmental conditions become more challenging. Conservation cloning offers hope for preserving genetic diversity in endangered species and potentially restoring lost biodiversity. As genetic analysis techniques become more sophisticated and less expensive, scientists may be able to recover viable genetic material from museum specimens, expanding the genetic diversity available for species recovery efforts. The emerging field of synthetic biology, which combines cloning with genetic engineering, could produce organisms designed to address specific environmental challenges. Researchers are exploring the possibility of creating microorganisms that could clean up pollution, produce sustainable fuels, or manufacture valuable materials through biological processes. Advances in automation and artificial intelligence are likely to make cloning procedures more efficient and reliable. Robotic systems could perform the precise manipulations required for nuclear transfer with greater consistency than human technicians, while AI systems could analyze embryo development and predict success rates with greater accuracy. The cost of cloning procedures continues to decrease as techniques improve and equipment becomes more widely available. This trend could make therapeutic applications more accessible to patients and conservation applications more feasible for protecting endangered species, expanding the beneficial impact of cloning technology. International cooperation in cloning research is likely to expand as scientists recognize the global nature of the challenges they're addressing. Climate change, infectious diseases, and biodiversity loss require coordinated responses that transcend national boundaries. Cloning technology could become a tool for global cooperation, rather than competition. Educational applications of cloning technology could help train the next generation of scientists and medical professionals. Students could work with cloned cell lines and tissues to learn about development, genetics, and disease processes in ways that wouldn't be possible with traditional educational approaches. Ethical frameworks for cloning applications continue to evolve as technology advances and societies gain experience with its use. Future policies are likely to be more nuanced and specific, distinguishing between different applications and incorporating lessons learned from current research and clinical experience. The integration of cloning with other emerging technologies could create possibilities that are difficult to imagine today. Combining cloning with advances in tissue engineering, gene therapy, and regenerative medicine could produce treatment approaches that address complex medical conditions in fundamentally new ways. Public understanding and acceptance of cloning technology may increase as people see beneficial applications in their own lives or communities. Successful treatments for serious diseases, effective conservation of beloved species, or solutions to environmental challenges could shift public perception from caution to support. As our gentle journey through the world of cloning draws toward its close, we arrive at perhaps the most beautiful realization of all. Despite our ability to create genetic copies, nature ensures that no two living beings are ever truly identical. Even clones sharing the same DNA blueprint become unique individuals shaped by the subtle influences of environment, timing, and the beautiful randomness that characterizes all life. This individuality emerges from the very first moments of development, when environmental factors begin influencing how genes are expressed. The temperature in an incubator, the nutrients in culture medium, the timing of developmental signals, all contribute to creating subtle differences that ensure each cloned individual develops their own unique characteristics. Consider CC, that first cloned cat we met earlier in our story. Despite sharing identical DNA with her genetic donor, CC developed a completely different coat pattern due to the random way X chromosomes are inactivated during female development. This discovery helped scientists understand that cloning produces genetic copies, not photocopies. Individuals who share the same blueprint but interpret it in their own unique ways. The study of identical twins has reinforced this understanding. Even twins who develop in the same womb at the same time, with the same nutrients and environment, emerge as distinct individuals. Their fingerprints are different, their personalities develop along separate paths, and their life experiences create increasingly unique identities as they grow and mature. This natural protection against perfect copying serves important biological purposes. Variation, even among individuals with identical genes, provides resilience against disease, adaptability to changing conditions, and the diversity that makes life both robust and beautiful. Nature seems to have built safeguards against the very uniformity that early critics feared cloning might create. The realization that clones become unique individuals has profound implications for how we think about identity and human value. It suggests that our worth doesn't lie simply in our genetic codes, but in the complex interplay of biology, experience, choice, and chance that shapes each person's journey through life. Cloning doesn't diminish human uniqueness. If anything, it helps us appreciate the many factors beyond genetics that make each person irreplaceably individual. Research into epigenetics, the study of how genes are expressed rather than just what genes are present, has revealed another layer of individuality that emerges even in cloned organisms. Environmental factors can cause chemical modifications to DNA that change how genes function without changing the underlying genetic sequence. These epigenetic modifications can be influenced by diet, stress, exercise, and countless other factors, creating another source of individuality among genetic copies. The peaceful acceptance of this natural variation has helped resolve many early concerns about cloning. Rather than producing armies of identical beings, cloning creates genetically related individuals who develop their own personalities, preferences, and paths through life. The technology respects rather than violates the natural tendency toward individuality and diversity. This understanding has also influenced how scientists approach cloning research and applications. Rather than seeking to create perfect copies, researchers focus on preserving valuable genetic resources while acknowledging that each individual, whether naturally conceived or cloned, will develop their own unique characteristics and contribute their own gifts to the world. The applications of cloning that have proven most successful and beneficial are those that work with rather than against these natural tendencies toward variation and individuality. Therapeutic cloning uses genetic copying as a tool for creating compatible tissues while respecting the fundamental humanity of all individuals. Conservation cloning preserves genetic diversity while acknowledging that each individual animal will adapt to their environment in their own unique way. Perhaps most beautifully, cloning has helped us appreciate the miracle of natural reproduction and development. By studying how genes are expressed and how individuals develop from single cells, scientists have gained deeper appreciation for the complexity and elegance of the natural processes that create new life. Far from diminishing the wonder of biology, cloning research has revealed new layers of beauty and complexity in the systems that govern life itself. As we've wandered together through the fascinating world of cloning tonight, we've discovered a story that's both more complex and more beautiful than the dramatic portrayals often found in popular culture. From the ancient dreams of creating perfect copies to the modern reality of using genetic technologies to heal and preserve, cloning represents humanity's ongoing conversation with the fundamental mysteries of life itself. The science of cloning reminds us that the most profound technologies often work by understanding and cooperating with natural processes rather than opposing them. As researchers continue to refine these techniques and explore new applications, they do so with growing appreciation for the complexity and wisdom built into the biological systems they study. Whether growing tissues to heal human disease, preserving the genetics of endangered species, or simply marveling at the intricate dance of cellular division, cloning science proceeds with the patient reverence appropriate to working with the fundamental processes of life itself. Until we meet again in these quiet hours, may your dreams overflow with the poetry of cell division and scientific wonder. And may you sleep knowing that even in a world of perfect genetic copies, your own journey remains beautifully unique.